Hi, everybody. Thanks first to the organizer for inviting us and for all of you who came to listen to this talk. I'm Daniele Armaleo, and this work was carried out by Lily Xiao and myself while she was an undergraduate in my lab at Duke University. She's now in grad school. So let me go to the slides. So we will show that lichens developed an unusual strategy to protect themselves against desiccation damage. And this strategy involves their ribosomal DNA introns. For that, we needed the help of yeast, Saccharomyces cerevisiae, who is a much more versatile organism to work with in the lab than lichen mycobionts are. However, the first experiments we will present was done with the lichen mycobiont from Cladonia grayi. And because we're talking about rDNA introns here, I showed you the the constitution of the Cladonia grayi that we used. The constitution of their ribosomal DNA repeats. And you can see by the colored boxes in the rDNA genes, the 18S, 5.8S, and 25S genes, that they contain a lot of introns, the majority of which are group 1 introns, which are the yellow boxes, and a minority are represented by spliceosomal introns, which are the orange boxes. The main characteristic of all these introns is that they cannot self-splice. Normally, group 1 introns found in most microorganisms and in their ribosomal DNAs are, first of all, fewer than in lichens, but the essential difference is that most of them can self-splice. What that means is that when the intron is transcribed into RNA, it folds into a ribozyme structure, which has the catalytic activity of splicing the intron out rapidly from the RNA. Lichen group 1 introns lack the sequences necessary for this catalytic activity, so they must need a machinery provided by the mycobiont that splices them out. The fewer spliceosomal introns that are in lichen rDNA are also an anomaly in the sense that they're in the wrong RNA, so to speak. Most spliceosomal introns are, of course, part of mRNAs that all eukaryotic have, meaning all eukaryotic have mRNA with introns, including fungi and lichens. So anomalously, lichens also have some of spliceosomal introns in their rDNA. So a common property of these introns, both the spliceosomal and the group 1 introns, is that they cannot splice themselves out, but require machinery. A spliceosome in the case of the spliceosomal, which all eukaryotic cells have, and a special machinery invented by lichens that only lichens have for the group 1 introns. So the core issue that we're going to deal with, and that we think is at the origin of all the phenotypes that we're going to talk about, primarily desiccation resistance in lichen fungi, is the lack of self-splicing ability of these introns, which results in interference with the ribosomal assembly, which is depicted in this slide. So when this slide depicts only the 18S region of the rDNA gene, and when that is transcribed, indicated here by this white little RNA polymerase, forming RNA, immediately that RNA is assembles with many protein factors, ribosomal protein, and assembly factors that fold it and interact with all these proteins while it is being transcribed and processed during transcription. The part that ends up in the ribosome at the end is this part in color here, which is the 18S region, whereas the 5' ETS and the 3' ITS1 are removed during processing. But if you introduce in this happy process the presence of introns in the DNA, when they're transcribed into rRNA, into ribosomal, into RNA, they changed the structure of this RNA, and therefore the folding interfered with the folding. So that would create a problem for the assembly of these particles, unless these introns were spliced out, which they in fact are, because ribosomal RNA, mature ribosomal RNA of lichens is devoid of any introns. But who's got to splice them out is the machinery, which is spliceosome in case of the spliceosomal introns and special machinery in case of the group 1 introns. But nevertheless, that machinery would also interfere with the assembly process. So the basic point that we're trying to make here is that this lack of self-splicing requiring machinery causes problems in ribosomal assembly, which are the beginning of the story here. Here, this is an outline of what happens normally in assembly with the best studied fungus, which is yeast. But it's also the case for many other fungi that have been analyzed. And that is that when the assembly proceeds normally and yeast is happy and grows fast, the RNA region around the 18S are removed very quickly, forming an almost mature 18S very rapidly, which then continues and gets assembled in the 18S subunit very quickly. However, when yeast is stressed out by environmental stresses or nutritional stresses of various kinds, the processing of the RNA is slowed down, and it's different in the sense that the 5' ETS is not removed immediately, and the 3' ITS1 is also left intact. What that does is that it stalls assembly here, because assembly cannot go beyond the initial stages unless these extra parts are removed. So the presence of an RNA, the lingering of an RNA intermediate that has these parts, is an indicator of ribosomal assembly stress. So to test that in lichen mycobionts, we used as a made a northern blot, and we used as probe the internal sequences of this third intron from the left here, which is a group 1 intron, to test what happens to the ribosomal RNA processing when we probe it with an intron probe. And here's the result of that northern. This is the gel in which we ran the RNA from the lichen mycobiont, in which you can see the 2 mature ribosomal subunits here. And then when we transfer that to a membrane and probe it with that intron probe, you'd see that there are 2 bands appearing. One is the size of the spliced out intron, 300 base pairs, which is a good thing because the introns eventually are all spliced out. But there is also the appearance of this 23S bands, which is the unprocessed part of the 18S RNA, which contains the intron because it reacts with the probe. And so in the lichen mycobiont, the presence of this also indicates that the ribosomal assembly process is slowed down. So for our yeast experiment, we used CRISPR to introduce an intron from a lichen, Cladonia grayi, and see what effect it has on growth and desiccation resistant. The intron that we use in this case is not a group 1 intron because yeast does not have the machinery to splice it out, and that would be lethal. But it's a spliceosomal intron, this one here, for which yeast has the machinery to splice it out. So we created 3 mutants, one with the intron, with the spliceosomal intron in the small subunit gene in all repeats. Here I only indicate 2. Another with the intron in the large subunit in all repeats, and another one with the intron in both subunits in all repeats. And then we tested growth and desiccation resistance. You can see here that the effect of growth is dramatic. This is the wild type without intron. And the cell duplication is dramatically slowed down in the intron-containing mutants. Here is the one with the intron only in the small subunit. Here is the one with the intron in the large subunit. And here is the one with the intron in both subunits. And you can see that there is dramatic slowdowns in growth. We were, however, amazed to find out that also desiccation resistance is dramatically affected. In this graph, you can't even see how many wild type cells resist desiccation. And you see that they increase in the intron in the small subunit, intron in the large subunit, and intron in both subunits dramatically, up to about 1700-fold higher desiccation resistance than the wild type. So we think, therefore, that delays in ribosomal assembly caused by delayed intron-splicing lead directly to slow growth and desiccation resistance, and I'll briefly outline how we think that that happens. Just a reminder that there are two kinds of introns, the spliceosomal introns in red and the group 1 introns in yellow here in lichen mycobionts. So let's see what would happen in an abstract case in the splicing of these introns out of the rDNA as it's rRNA as it's being assembled, as indicated here. So let's start with what the spliceosomal introns do. So this would be one intron here, one spliceosomal intron, and as it's being assembled into the subunit and then spliced out. So they require spliceosomes from the cell, and many of them have to be recruited to splice out all the introns in the many, many repeats of rDNA. So all of a sudden, the number of repeats is the number of spliceosomal introns in yeast is dramatically increased by the presence of this one in the rDNA. And so they basically suck up a lot of the spliceosome from the splicing process that is necessary to process the mRNAs that encode for other proteins, that encode for proteins. And the majority of RNAs in fungi with introns happen to be ribosomal protein genes. And so what that does is when the spliceosomes are basically hijacked by the rDNA splicing process, there are fewer of them available to make ribosomal proteins, and therefore there will be fewer ribosomal proteins. Let's suppose that now this is not a spliceosomal gene, but a spliceosomal intron, but a group 1 intron. It also has to be spliced out by a machinery provided by the microbiome. But in this case, the machinery does not interfere with the ribosomal protein, but it interferes with ribosomal RNA processing and therefore slows it down. And so the combination of fewer ribosomal protein and fewer ribosomal RNA made in a given time results in fewer ribosomes. And fewer ribosomes in the cell would have a dramatic effect on overall biochemistry. The obvious one would be slowing growth. A less obvious one is an effect on desiccation. Why would they increase desiccation resistance? For that, briefly, I will mention that the crowdedness of protein in the folding and processing behavior of proteins in the normal cytoplasm is crucial. Proteins are very concentrated, present at very high concentration in cells, and they have to sort of negotiate a very fine line between folding property and folding improperly. And every time the concentration increases, that is has a dramatic effect on folding and aggregation of proteins, which get damaged, and there is phase separation, and the defenses of the cells are overwhelmed, and also the ribosomes get damaged. In a cytoplasm, which is a diluted soup of a normal cytoplasm, like it would happen in this case, all these effects are delayed because the aqueous volume is larger, there are fewer proteins, there is less misfolding, less aggregation, less phase separation, lighter load on induced defenses like heat shock proteins, and less damage to ribosomes, all of which we think leads to increased desiccation tolerance. So the things that I leave out here, I just will briefly mention, are the fact that this ribosome drop in concentration is a constitutive defense because this is caused by the constant operation of these introns whenever rDNA is made, or RNA is made, which is, of course, always. But there are many other defenses that the cell use that are induced by desiccation. And so these are different from the constitutive defenses like the ribosome drop caused by introns. In yeast, there is also another difference, another properties that you have to look at to consider its desiccation resistant, and that is that it is stochastic. Very few cells are desiccation resistant. Most of them, when the cells are desiccated, die, and only the few that survive will then reconstitute the population. In lichen, desiccation has to be constitutively expressed in all cells because lichens can't afford to lose most of their thallus whenever they desiccate, which is a lot. And therefore, we think that these introns provide the constitutive background against which makes all the cells of the thallus resistant to desiccation. So this is the sort of the end of the talk, and I would like to acknowledge the funding sources, which are intramural funding from Duke University and crowdfunding from experiment.com. And that is the end of this talk. Thank you very much. And I now go back to the regular screen and take any questions if you have any.